At earlier you can see in the movie the actin look like it's moving, but it's not actually move by itself. It just change the plus and the minus end position. And the reason the for dissociation of actin at the minus end is because when the actin combine to form the uh, F actin, they are ATP actin. Just after around 2 seconds after incorporating into the F-actin, the ATP will be hydrolyzed into ADP and phosphate group. However, the phosphate group will not be released from the actin until 50 seconds later. And once it's released, the ADP actin will have a tendency to dissociate from the F-actin. And based on the critical concentration, we have the Treadmilling range in the range of 0.12 to 0.6. In this range of concentration, the minus end will be shortened while the plus end will be elongated. And as you can also see in here, the elongation rate of the plus end is much higher than the minus end. And the dissociation rate also higher at the plus end compared to the minus end. Um, this concentration actually much lower than the actin concentration inside the cell. Hopefully you can still remember that in the normal cells, the actin concentration is in the range of around 0.1 to 0.4 millimolar. So it's higher than critical concentration. However, the inside the cells, the actin is rarely randomly form or grow. And the reason is because even though there's a lot of actin inside the cells, those actins are bound by something else. Also, we have profilin. And earlier, it's promoting the exchange of ATP for ADP and lead to formation of ATP actin. So this ATP actin is, in usual case, it can involve in the formation of new actin. However, profilin will prevent the random association between the ATP actin to form nucleus, and therefore it prevent random formation of actin. Can only it facilitates the formation of actin by interacting the formin at the plus end. And the other protein is a coffulin. Coffulin function is to sever the actin F actin or actin filament into shorter fragment and then dissociate the G actin from F actin, and this to provide keep providing a new pool of the G actin for uh, growing at the plus end. The other protein is thymosin beta 4. Thymosin beta 4 is bind to uh, the ATP actin and prevent the ATP actin involving in the polarization of F actin. So, so that the available ATP actin is depends on concentration of profilin as well as the concentration of thymosin beta 4. Some actin which are required to be more stable, or sometimes they may not, but most of stable actin will be kept by protein at the plus end by the cap G. It bind to the plus end and it prevent the G actin from dissociating from the plus end, as well as prevent the new G actin to combine to the associate to the plus end. So if the cap G bind to plus end, the plus end will not change. It's stabilized. And troponodylin bind to the minus end. Troponodylin probably I say about it. Troponodylin bind to to minus n, and when it bind to minus n, it also stabilize the minus n. So you could expect that if the actin with tropomodulin at the minus n and CAPG at the plus n could have the stable length. The length will not change. So for those actin which require to be very stable, such as the actin in our muscle, it requires another protein called tropomyosin. It binds along the length of actin, and this tropomyosin have two main functions. One is to stabilize F-actin. The other function is to control the interaction between F-actin and myosin. F-actin and myosin. So, are there any stable actin structure which we could expect those stable actin structure would have at least these three proteins: the CAPZ at the plus end, tropomodulin at the minus end, and tropomyosin along the length of actin. So the actin inside the cell, they do not exist as a single filament, but they usually combine together to form bundle. And one of the protein that involved in the bundling actin are called as the actin cross-linking protein. So one of them is fibrin. So fibrin have two actin binding domain in blue and calcium binding domain in purple. So because it had two different actin binding domain, it can bind to two different actin at the same time, and to form together they will form the parallel network, parallel bundle of my actin. And this fibrin can be observed in microvilli, in filopodia, as well as in focal excitation of the cell. The stereocilium, the cilium is usually the cilium is the name for microtubule supported structure, but stereocilium is structured quite large. So at first people think that it's a cilium, but it actually is a microvilli. Okay? So the naming is wrongly. So stereocilium is actually a microvilli, and it is supported by parallel actin. It's supported by parallel actin, and those parallel actin are formed by fibrin. So a stereocilium is a structure inside our ear, in the ear. And another protein which can also lead to the formation of parallel bundle alpha-actinin. So alpha-actinin has to work as a dimer because they have only one actin binding domain. So it also form the parallel actin found in stretch fiber, in the filopodia, in muscle G line. So what the difference between these two? As you can see here, the distance between the two actin fiber is much larger than the alpha-actinin bundle compared to the fibrin bundle. And this large gap between two actins allow the myosin bundle or myosin fiber to be in here, and allow this bundle to contract. This the bundle generated by fibrin cannot contract. Only this one can contract. Right? So try to remember that. And other protein can also lead to cross-linking of actin is spectrin. Spectrin. Spectrin is quite long structure, and it's will cross-link short fragment of actin. This short fragment is around half of the turn. It contains 14 G-actin instead of 28 of the complete turn. So it's only around, let's say, 37 or 36 micrometer long. Other protein is spectrin is observed under the human red blood cell and under the red blood cell. It strengthens the red blood cell membrane. Filament can only can have it has two actin binding domain, and it bind to the actin in many orientations, and therefore it leads to formation of a meshwork, an actin meshwork, with no obvious organization of actin filament, and it's observed under in the cell cortex, just under the cell membrane. It's also observed in the leading edge of the cells, in the cell tribal and phylopodium. So the filament could generate the actin meshwork in I think in all of the cell type. Here is how the actin meshwork look like. We have no obvious or no dominant orientation of actin filament. And the other protein, cross-linking protein, is dystrophin. It connect the actin protein to the cell membrane, and disruption of dystrophin function could lead to dystrophin, muscular dystrophy. Um, there are some other proteins can help the actin uh, to bind to the membrane or protein on the membrane. So we have spectrin in the red blood cells and just under the membrane, cell membrane with red blood cell. It can have the actin to bind indirectly to the band 3 protein on the cell membrane, and the actin can interact with the band 4.2 and indirectly to take the actin to glyco 14C on the cell membrane. And here is a stereocilium. Stereocilium. Here we have multiple with stereocilia. So these are newly formed stereocilia, and we know that when the stereocilia elongate, it should be elongate at the plus end. So this is the plus end of the actin should be somewhere here. And this can actin, the parallel actin can link to the cell membrane protein like E-string or and EBP15. It can be observed in the microvillus as well as the stereocilia. So remember that the stereocilia is actually a microvillus, it's not a true cilia. Um, involving in generation of actin or actin polymerization, there are two important proteins. One is formin and another is ARP2 and 3 complex. So the forming on work at the dimer. It's on the way work at the dimer. It has some important domain like the regulatory domain, where it bind to a small GTPase and it affect the activity of the formin. It has the formin homology domain, FH1 and FH2. There's two domain. One domain is interacted properly, and therefore it bring the actin into the FH2 domain. So here it's the structure of FH2 dimer. So FH2, so we have the FH2 should be here. This one should be the F81. So the F81 interact with the porphyrin and bring the actin to close vicinity of the F82 domain. And then F82 domain will attach the actin together and to form the F-actin. So as you can see here, the F82 domain always attached to actin with incorporated to plus n, and therefore the formin always stay at the plus n, the actin. I think probably I can go to the first slide and let you see the movie.
Bundles and branching is particularly important engine by the mechanical membrane for cell movement at the plasma
So
membrane.
here is how this is the formin. It should be the F82 domain. And this is F81 domain. And this is the actin filament. So as you can see, the F81 domain receive the propylene together with actin to bring the actin into F82 domain.
For cell movement at the plasma membrane.
Hey, oh, sorry, I forgot. Let me reshare the slide. Okay, so that is how the forming work. And the forming in normal situation is inactivated by folding on itself. And when the row binding domain bind to row GTP, row is a small GTP. So when row bind to GTP, it become activated, and then it bind to row binding domain of forming, and then activate the forming. So one forming is activated, it's dimerized, but in here they draw only one. And it receives the actin porphyrin by the F81 domain and bring the actin to F82 domain to generate a new actin. And because the draw GTP is attached to the cell membrane, for the formin always be activated close to the cell membrane. And when the formin is activated, the new actin will generate close to the cell membrane with the plus and toward the cell membrane and the minus and toward the cell center. So here is the how the small GTP can be regulated. So the small GTP, they had GTP activity, and when it's hydrolyzed the GTP, it can hydrolyze GTP into GDP. So when the signal comes, the gap is activated. This is guanidine nucleotide exchange factor, and it could stimulate the exchange of GDP by GTP and therefore activate the Rho. Rho can activate formin, for example. And after some time, the GTP activating protein can bind to Rho GTP and activate the GTP domain of Rho, and therefore hydrolyze GTP to a phosphate and a GDP inactivated Rho. So remember the GTP small GTP protein only activated when it bind to GTP, and it become inactivated when it bind to GDP. Another protein called as guanine nucleotide dissociating inhibitor and bind to this complex and prevent the exchange of GDP to GTP and therefore keep the raw protein in the inactive form. And in the cell, when Rho is overactivated, it leads to formation of stretch fiber. So you know that because Rho, when Rho is activated, then formin is activated, and formin will generate the actin, the unbranched actin, and those actin will combine together by alpha actinin to form the stretch fiber or parallel actin fiber. The other protein important for actin polymerization is ARP2 and 3 complex. It include one actin-related protein 2, actin-related protein 3. They have 3D structure very similar to actin. Yeah. And there are some other accessory protein. So the ARP2 and 3 complex actually it works at the nucleus for actin formation. It works at the nucleus for actin formation. Its activity is regulated by a nucleation promoting factor. Nucleation promoting factor 3 have the three different domain like uh, acid domain, connecting domain, and warp homology domains. So when the nucleation promoting factor is activated by other factor, it can bind to actin and then it's bind to ARP2 and 3 complex. So ARP2 and 3 complex with a nucleation promoting factor then can bind to the available actin at a certain angle, and one is bind to another actin, then ARP2 and 3 complex is fully activated, and it attach a new actin to the ARP2 and 3 to form new F-actin. And because the up2 and 3 complex have to bind to available actin to be activated, fully activated, it will generate uh, the network of actin which are linked together by up2 and 3. And it look like a tree, and this actin are called as a branch actin, and it has a T shape with many branch at the top. And nucleation factors are regulated in here by CDC42. CDC42 is also a small GT base. It belongs to the Rho family, and it combines to the Rho binding domain of the nucleation factor. And beside, they have the basic domain which bind to uh, PIP2 on the cell membrane, and it leads to the formation, the activation of nucleation promoting factor, and lead to activation of up2 and 3 complex, 2, 3 complex. So because all of these are membrane component, up2 and 3 complex, similar to Fogmin, always be activated close to the cell membrane. And it from here you generate the new actin with the blood end pointed toward the cell membrane, and the minus end stay at the up2 and 3 complex. So compare the actin and Fogmin and up2 and 3 complex. Uh, Fogmin generate actin, facilitate the polymerization of actin at the plus end, while the up2 and 3 complex serve at the nucleus and promoting formation of actin from the minus end. And up2 and 3 complex could lead to formation of lamellipodium, with a network of branch actin at the leading edge of the cell. At the leading edge of the cell, when uh, nucleation promoting factor is activated, up2 and 3 complex can bind to available actin and fully activated to generate new actin. So this new actin point the plus end toward the cell membrane and probably involved in pushing the cell membrane forward. And further at the back, some F-actin could not attach to the cell membrane by some other protein. Then it will not generate any force to push the cell membrane forward, and therefore they could be kept by the cap jet. And then when the cap jet bind to plus end of this actin, it stop growing. And after some time, when the ATP is hydrolyzed into ADP, then coupling can bind to ADP F-actin and dissociate the G-actin from the F-actin, and those G protein would be transported toward the leading edge and involved in the formation of new actin. In bacterial, they have a protein, a nucleation promoting factor that can activate up2 and 3 complex. And when the up2 3 complex activated on the bacterial cell membrane, then the formation of actin in gray here can push the bacterial forward. This is a electron microscope image, and the black dot here is the bacterial, and this is the actin filament formed by activation of the up2 and 3 complex. And this is how it look like under fluorescent microscope. We have a bacterial inside another inside an eukaryotic cell, and bacterial is in red while the actin tail is in yellow. And the reason for it, because like on the Listeria monogenesis bacterial can cause a Listeria disease, it had a protein called ACA. So ACA is nucleation promoting factor. And ACA can activate up2 and 3 close to it. And the up2 3 can lead to formation of new actin and push the bacterial cell forward. When the actin and in the cell, the up to 3 complex also interact with the VAS, and VAS would keep actin can be added here and prevent the CAPZ to bind to blood and so when the CAPZ bind to blood and it will prevent the formation of new actin, and this new actin later be dissociated to provide G-actin for elongation of actin here to push the bacteria forward. I think it would be easier for you to watch the movie.
This mammalian cell has been infected with pathogenic Listeria monocytogenes. These bacteria move throughout the cytosol by recruiting host cell actin, which polymerizes and pushes them forward, producing a comet's tail in their wake. Whenever a bacterium is pushed into the plasma membrane, it creates a temporary protrusion and is then bounced back to continue its random path.
So in here, the bacteria in
If we
black.
look closely, we can see a bacterium divide inside the host cell. Immediately after separation, the two daughter cells assemble their own actin tails and start moving about.
Okay, so because of the
These
actin,
bacteria can also form actin comet tails and move in cell extracts. Here, the bacteria are expressing the green fluorescent protein, and actin is labeled red with a fluorescent dye. The dynamics of the actin tails that propel the bacteria through the cytosol can be modeled based on known biochemical and physical properties of actin and actin filaments.
okay, so on iBiology channel on YouTube, there are two great lectures by Julia Theriot about the actin and dynamic of actin. You can watch them. If you cannot find them, then tell me. I can send you the link. So they study the movement of Listeria cytogenesis by actin polymerization, and when she scales the size of bacterial as well as the scale the size of actin, so that the bacterial at large and the submarine, then the polymerization of actin can put that submarine down at a speed around 50 miles per hour, very fast speed. Okay, and this up to in 3 complex also involves in the formation of endocytosis, and it's also involved in the movement of those endosomes, like transferrin is a protein involved in absorption of the iron from the blood into the cell. So it's small when it forms small vesicle, the nucleation promoting factor can activate the up to in 3 complex and lead to formation of actin and allow those vesicles can move in the cell. So the activation of up to in 3 complex lead to the formation of new actin to push the cells or to put the vesicle forward. That you can be utilized by bacterial cells as well as endosomes in our cells. It's also involved in phagocytosis when bacteria are recognized by antibody, and the leukocyte with the receptor on the antibodies on the cell surface attack the bacteria, and then the up to 3 complex is activated and lead to the formation of the phagosome. Protein that interact with actins is myogenes. In human genome, there's around 45 genes encoding for myogenes, and by using the sequence of S1 head domain of around 40 myogenes, they can draw this phylogenetic tree to show the relationship in evolutionary relationship among different myogenes. For this, some of these myogenes, the mutation could lead to deafness or blindness. So among those myogenes, there are myogene 1, myogene 2, and myogene 5 are the 3 most studied myogenes. So myogene 1 work as monomer of heavy chains. So it had one heavy chain with the head domain had the actin binding domain, and it also had the actin ATP binding domain in here. And it had a neck where some light chain can bind to it. So light chains are different polypeptide, and the tail. The class 1 had the tail can bind to the membrane and bind to membrane, and it can involve in the movement of small vesicle, then also the movement of the membrane. In human genome, there's 8 genes encoding for the myogene 1. Myogene 2, they have 2 heavy chains. They work at the work at the dimer of heavy chain. Each heavy chain had a head, neck, which is usually bound by 2 other light chains and the tails. The myogene 2 is usually bundled to form a bipolar myogene 2 bundle with the head at the both ends of the bundle. And this bundle will combine with the actin to form the sarcomeres or to form the muscle inside of our body. It's involved in the contraction of our body, movement of our body. And in human, there's 40 genes encoding for myogene 2. Myogene 5 also had 2 large heavy chains and multiple light chains binding to the neck of the heavy chain. And the tails and the cargo interaction domain allow the kinesin 5 to attach to small vesicle or small organelle inside the cells, while 2 heads allow the myogene to move on actin. And this movement can, because it had 2 different heads at any time, it only attach to at least 1 head attached to actin, and therefore it can work on actin and involved in organelle transport, especially in fungi, in plants, fungi and algae cells. Okay. Probably we can stop here and next week we continue. Okay. So some of you may save my number, and if there's any kind of disconnection, please call me. Okay.
Yes, I got it.
Do you have any question for the part we have studied so far?
Đến giờ thì chưa ạ. Bọn em sẽ về đọc lại một lần nữa, rồi có gì không hiểu bọn em sẽ hỏi thầy sau ạ.
Ok. Thế thì chúng ta nghỉ ở đây nhé.
Dạ vâng ạ.
Chào các bạn.
Em chào thầy ạ.
Chào thầy ạ.
Em chào.
Chào thầy ạ.